So this Cultivating Healthy Plants webinar series concerns integrated pest management, and it is a collaborative effort between the University of Idaho, Montana State University, University of Nevada, Reno, and Utah State University. All of our extension programs come together to host this monthly through the academic year. My name is Desiree. I'm the program manager for the University of Idaho's Extension IPM program, and I am the host for this round of our Cultivating Healthy Plants series. Today, I'm excited to let you all know that Andrew will be chatting with us about how insects work and how to manage them. As an entomologist, I'm totally biased and excited to hear this talk. All right, so just a few Zoom logistics before we get started. You can exit full screen if you came into it by looking at this view options and exiting full screen, or oftentimes you can just hit your escape key if you seem to be locked into full screen. And then there's two similar looking icons on your Zoom page. One of them says Q&A and one of them says chat. And this is important because we're going to be using them differently. The Q&A, we're probably going to just have Andrew address questions that you have at the end of the webinar. So this is questions about the content. However, if you have sort of logistical issues or technical issues, hopefully you can send something to the host and the panelists, and Carrie and I will help field those questions or issues during the webinar. Note that if you are looking for CEUs at the end of this webinar, the chat section is where you will find a link to a survey that we're going to ask you to take to give us some feedback, and then we'll also run you through how to go about getting those credits. Furthermore, if you would like closed captioning, that has been enabled, and there's a little CC icon also on your Zoom tools menu that will allow you to do that. Next, another little housekeeping item is I am showing you this to let you all know that this is a federally funded program. And since we received that federal grant funding, we're showing you this poster to inform you that all of our universities and extension parts of our universities are responsible for implementing the requirements of equal opportunity, and we do so. And with that, I will turn it over to Carrie to introduce Andrew.
Thanks, Desiree. My name is Carrie Jensen, and I'm the IPM program coordinator at the University of Nevada, Reno's Extension. And I'm very excited today to introduce our speaker, who is from UNR. Dr. Andrew Nuss is an assistant professor in the Department of Agriculture, Veterinary and Rangeland Science at the University of Nevada, Reno. He has a bachelor's and master's degree in entomology from Purdue University and a PhD in entomology from the University of Georgia. His lab at UNR investigates the physiology of insect neuropeptides and their roles in insect behavior, digestion, and nutrient storage. And his work serves as a basis for discovering new pesticides and insecticides. And his presentation today, he'll give us an overview of insect physiology, i.e. how insects work, and how different insecticides work to manage them. With that, I'll hand it over to you, Andrew, to share your screen and get started with this great presentation. Thank you for being here.
All right, thank you very much. Let me just share and we'll get started here. Okay, there we go. Yeah, thank you for that kind introduction. As noted, I'm well, now an associate professor of entomology in the Agriculture, Veterinary and Rangeland Sciences department at the University of Nevada. And I study a number of different things about insects and other arthropods. Some major areas we focus on are mosquito olfaction, to understand why certain mosquito species prefer to feed on humans versus other animals and how we can deter them from that. But also, as noted, I work on insect neuropeptides, particularly in this insect, Lygus hesperus, the western tarnished plant bug. I'll try to talk a little bit about that at the end. And although we want to understand what is going on with neuropeptides on a basic level in these insects, these can be targets for new insecticides. But overall, I focus on insect physiology, and that is, how do insects work? So it's a good basis for my talk today, how insecticides work. Okay. So I don't know why your background, why you're interested in today's topic, although I suspect a lot of you are interested in gardening. But I'm going to go fairly broad in this talk, covering some pesticides that are commonly used in urban pest management, others that are used for row crops, others for vector management. So I'm going pretty broad. I'm also going to talk about a few pesticides that are no longer in use. So they do serve as good examples, but I don't want that to be confusing at all. And I realize it may have been a while since the last time you all have taken a biology class. So I'm going to do a little bit of review at the beginning, or at least overview, and talk about insect physiology. So insects are animals like us, and they help to solve a lot of the same problems that we do. You know, they need to acquire food, they need to extract nutrients from it, and they need to expel waste. They also need to breathe to acquire oxygen so that they can utilize the food that they've acquired, and they need to grow and reproduce. So insects have solved some of these problems the same ways that we have. For other things, they've taken a very different path. So I'm going to give you this very brief overview of some of the major organ systems in insects, just to give you context for what's to come. So I'll start out with the nervous system. The insect nervous system is it's organized a little differently from ours. They have a brain and several ventral nerve cord ganglia, but in function, the general principle is the same. They're going to perceive stimuli from the environment, such as taste, touch, smell, sound, and sight. They're going to process that information and coordinate movement of the body in response. So we'll get into the specifics of how neurons work, because many insecticides do target the nervous system. Insects also have a digestive system. That's probably not too surprising to you, since most insect damage is caused by their feeding behavior. Now, the digestive system is organized a little differently than our digestive system, but the general purpose is the same: to digest food and absorb ingested nutrients. Insects also have an excretory system to eliminate wastes. This is primarily through the Malpighian tubules that are attached to the digestive tract. Roughly, these are somewhat analogous to our kidneys. Insects also have muscles. Again, not too different from ours. They largely function in the same way, though, of course, they attach to the body a little bit differently, just because the insect body plan is pretty different from ours. Insects also have an endocrine system. So there are several organs or glands that produce and release hormones that coordinate different physiological activities. So there are hunger signals, for instance, or reproductive hormones, or molting hormones, and so on. And scientists have taken advantage of insect hormones and have developed certain insecticides to target these hormones, which I'll talk about a bit. So one very major difference from us and between us and insects is that insects have an exoskeleton. That is a hard cuticle that supports their bodies and provides a point for muscle attachment, but also provides protection from the environment. And this is obviously from physical protection from injury, but the exoskeleton also protects the insects from desiccation. There's a thin waxy layer over the surface of the exoskeleton that prevents the insect from drying out. And this is really important because insects have a very high surface area to volume ratio. They lose water much more easily than we do. So this exoskeleton is produced and maintained by a layer of epithelial cells, essentially the skin of the insect, but that's underneath the exoskeleton itself. All right, insects also need to breathe. They use oxygen just like we do, but they've taken a very different tack on this. Instead of breathing through their mouth, they breathe through a series of trachea that radiate throughout the body. You can see that on this insect, these kind of white tubes that are going throughout this caterpillar, and air enters the body through little holes in the sides known as spiracles. All right, insects also have a circulatory system that transports nutrients, but not oxygen. Again, they don't have lungs like we do. But also, unlike our circulatory system, the insect circulatory system is an open circulatory system. They don't have vessels and veins, but they do have what's known as a dorsal aorta. This is a tube-like organ that is somewhat analogous to our heart and pumps fluids throughout the body. We refer to insect blood as hemolymph. Okay, moving on to reproductive organs. So in insects, males have testes, females have ovaries, not too different from our reproductive system. There are some variations and differences, but in general principle, the reproductive system is somewhat similar. Okay, so insects also have storage organs for provisioning nutrients. Primarily, this is the fat body. This is a kind of decentralized tissue system. You could call it an organ, but it's mainly similar to fat tissue in our body with some attributes of our liver also. Okay, so I know that was a whirlwind tour of insect physiology in just 5 minutes, but I did want to give you a brief overview because I will talk about some of these systems in more depth. And I also want to note that together, these systems are all working to promote survival in the organism. And a disruption of any one of these systems could result in the death of the insect or some other unfavorable outcomes to them. So these are weak points in insect physiology. And we found some ways to interfere with these systems, mainly chemically, which I'll be talking about today. So I'll mostly be focusing on the nervous system, also metabolism and the digestive system, and some aspects of the endocrine system, particularly as it relates to the formation of the exoskeleton. So I'll also be primarily talking about mode of action, or sometimes that's referred to as mechanism of action. Essentially, how does the insecticide work to kill the insect? So I've listed several modes of action on this slide that I'm going to try to cover today. This doesn't cover all insecticides, but it does cover a lot of the major ones. So among these, most current insecticides are nerve poisons. Now, before I get too deeply into that, I just wanted to share this poster from the Insecticide Resistance Action Committee, abbreviated as IRAC, not to be confused with the Middle Eastern country. But this poster details several different modes of action of different insecticides. So there are 36 classifications on this poster, plus a few miscellaneous. It's pretty expansive, and you're going to have to zoom in to read it. And even this doesn't include all modes of action. I recommend you look this up at some point in your free time through their website. But one thing I want to highlight is that this poster is subcategorized by color. And you'll note that blue here covers the nerve and muscle toxins with heavy emphasis on nerve, really. And if you let your eyes unfocus just a little bit and look at this poster, you'll notice that a large portion of this is actually blue. About half of these are nerve toxins. So there are several types of nerve toxins, and I'll be talking about two main categories: axonic poisons and synaptic poisons. So axonic poisons disrupt the sodium and potassium ion channels along nerve axons. So again, a little bit of biology review. When an axon, that is a nerve cell, is activated, an electrical impulse travels down the axon in the form of opening sodium channels. This isn't quite the same as an electric charge moving through a wire. That's the movement of electrons. But instead, in an axon, it's mediated by electrochemical gradients. So normally the nerve in a resting state has a negative charge internally. This is from chloride ions. And external to the neuron is a positive charge. This is from sodium ions. So opening of sodium channels in the nerve itself causes these sodium ions to rush in rather rapidly to the interior of the cell. Because as we know, positive and negative charges attract one another. So this change in charge activates the next sodium channel, and then that one activates the next sodium channel to open, and so on and so forth in a wave-like motion, a series of chain reactions moving down the cell. Think of like dominoes falling down, for instance. So afterwards, these channels close rather rapidly, and the nerve can reset itself, ready to fire again. Now, this may sound like a rather complicated process, but it obviously works, and it works quickly because that's how our neurons work as well. So axonic poisons disrupt these processes by either jamming these sodium channels open or keeping them closed. So this can cause the axon to fire continuously if they're held open, and can cause convulsions and paralysis and eventually death in the insect. The insect can't really do anything because it can't coordinate any of its actions. I mean, nerves are helping it move and interpret the environment. So this is disrupting that process. Or if the sodium channels are held closed, I mean, the insect is simply paralyzed. It can't move. It can't get the message to actually move. So if you look at a pesticide label, and you can find out what insecticide is included by looking at the active ingredient. And I've listed a few of these active ingredients for axonic poisons here, as well as what pesticide class they belong to. So some major examples of axonic poisons include pyrethroids, permethrin, lambda-cyhalothrin. There are many more examples. Organochlorines are axonic poisons. DDT is a major representative. Oxodiazines, indoxacarb, or semicarbazones such as metaflumazone. So now all of these different classes don't damage the axon quite in the same way. Some hold the channels open so the nerves continually fire until they give up and die. Others keep them jammed shut, preventing electrochemical transmission, but as a whole, they're interfering with nerve transmission. So these pesticides typically induce quick knockdown. They incapacitate insects rather quickly because they're halting the ability of the insect to move and respond to its environment. And you may notice, again, some of these insecticides are from a bygone era, notably DDT. We, by and large, don't use that anymore these days. But I've included older insecticides just because you may have heard some of the names before, and they're good examples. There are reasons why these were phased out, which I'm not going to get into today, but the class I highlighted here in orange, pyrethroids, definitely is. We use a lot of those nowadays. Okay, so in contrast to axonic poisons, we also have synaptic poisons, and these interfere with nerves at the junction of two nerves. So, you know, we don't, in our bodies, we don't have a nerve that runs straight from our brain all the way down to our toe. That would just be physiologically impossible for a nerve to be that long. So we have a series of nerves that interconnect in our body that transmit the signal from each other to other parts of our body. And in our brain, you know, we have billions of neurons interconnecting. So it's the same deal for insects, but there needs to be a way for this electrochemical signal to get to the next neuron, to jump the gap between cells. So this connection or gap between nerves is termed the synapse. And in these diagrams, it's called the synaptic cleft, same thing. It's a very small gap, but it still is a gap on a cellular level. So to bridge this communication gap, the activated neuron releases little chemicals known as neurotransmitters into the synapse. So that electrochemical signal I was just talking about comes barreling down the axon until, you know, bam, it hits the synapse, and these neurotransmitters are released into the synaptic cleft. So then these neurotransmitters bind to and activate receptors on the receiving cell. And then that cell starts opening sodium channels, and that nerve transmission can continue along in that receiving cell. So one very common neurotransmitter is acetylcholine, abbreviated as ACH in this diagram. And in a normal synapse, the neurotransmitter does its job, it transmits the signal, and then is broken down or reabsorbed by the cell more or less immediately. I mean, you don't want these neurotransmitters to persist in the synapse, or the axon is going to keep firing when it's not supposed to. However, many synaptic poisons inhibit the enzyme that breaks down acetylcholine, an enzyme known as acetylcholinesterase, abbreviated as ACHE. And that allows the acetylcholine to persist, and it keeps the nerve firing when it's not supposed to. So if you've ever seen an insect poisoned by one of these types of insecticides, you may see them undergoing uncontrollable tremors or convulsions. It's because they can't control their actions because their neurons keep continually firing. Eventually, these neurons continue firing, which leads to paralysis and death in the insect. So that's one way we can interfere with signaling at the synapse. But this can also be disrupted by interfering with the receptors of acetylcholine themselves, this ACHR receptor. And this can be disrupted by blocking the receptor. Some chemical may be able to bind to that so that acetylcholine can't fit into it and activate that receptor. Or some insecticides actually mimic acetylcholine and cause that receptor to fire when it's not supposed to. So either one of these actions is going to be detrimental to the insect and doesn't permit normal neuronal firing and eventually causes death. So a few examples of synaptic insecticide classes include organophosphates and carbamates. Chlorpyrifos and acephate are examples of organophosphates. Carbamates, carbofuran and carbosulfan. But these insecticides were more important in years past, but they do have some side effects on humans and other animals. So there's been an ongoing effort to phase these out. That's not necessarily complete, though. But the other major group I mentioned is chemicals that interfere with the acetylcholine receptor itself, either by blocking it or causing it to fire uncontrollably. Neonicotinoids are a somewhat newer class of insecticides based on the structure of nicotine. But as I was just mentioning, these interfere with acetylcholine receptors. And there are a few others, such as nicotine itself, pyridilidines, spinosins. Spinosins are bacterially derived insecticides. And there are a few other synaptic poisons that work similarly to the acetylcholinesterase inhibitors or receptor inhibitors. And I'll mention them very briefly. Several of these attack different types of synapses that use different neurotransmitters than acetylcholine, but the general principle is the same. So for instance, there are octopamine or tyramine receptors. Amitraz is an example of an insecticide that targets these receptors. This is a useful miticide. Several insecticides target gamma-aminobutyric acid or GABA receptors. Phenylpyrazole represents it is one class of these insecticides with fipronil as an example. Chlordane from the cyclodiene organochlorines is another older example, although this molecule is a bit more stable than in the environment than we generally tolerate these days. But there are some other assorted classes as well, such as metadiamides and isoxazolines. There are also what are known as chordodontal receptors in the insect. These are nerves that perceive sound. And these nerves are targeted by several classes of insecticides, including pyridines, fluindicimide, dipropyridaz, all of which disrupt nerve transmission. I don't want to get too far down into the rabbit hole of these, other than to say that they exist and target receptors in the nervous system, though. But this is just naming a few. There are other examples, and this may be too much of a wall of text already, and you may be glazing over. But this does help reemphasize what I said before, that axonal and synaptic poisons are among the most common types of insecticides. One thing I do want to mention is that this diversity of target sites does give us a lot of alternatives if insects do develop resistance, or if we continually rotate these different modes of action, we may be able to prevent insecticide resistance to take root in the population in the first place. But as far as downsides, axonic and synaptic insecticides have risks to humans and other vertebrate animals or non-target insects because we have axons and synapses as well. Now, the chemicals that are used for insect management were largely selected because they're more effective on the insect versions of these sodium channels or acetylcholine enzymes or receptors, but they can still have an impact on our physiology. All right, moving on, let's look at metabolic poisons. So I don't know if all of you recall what organelle is the powerhouse of the cell. Now, I hope you're all silently saying to yourself, the mitochondria, because I think that's the one thing everybody remembers from biology class. But this is the organelle that helps break down stored energy, such as glucose, into units that are usable by the cell, such as adenosine triphosphate or ATP. So these mitochondria are in every cell, and that's what keeps them going. So metabolic poisons disrupt these breakdown processes, including the electron transport chain and the respiratory enzymes that are part of that. And you can see in this diagram, several of these enzymes are part of this process. And we have several insecticides that target these particular enzymes. So when an insect is poisoned by one of these insecticides, essentially this process is disrupted and the insect keeps burning energy but gets none of the benefit from it, or is simply unable to access that energy that they have available. So the cells run out of energy and grind to a halt, essentially starving to death. So if we look at our modes of action again, notice that many of these insecticides are tinged with red, indicating that they target respiration, that is the process of releasing energy from stored food using oxygen through the mitochondria, about a third of these. And I've listed several classes of insecticides that target different parts of the electron transport chain. I'm not going to read off every one of these tongue twisters, but I do want to note that there are several different classes because, again, there are many different enzymes that are part of the electron transport chain and therefore many different potential targets. Now, a few that stand out to me are chlorphenapyr, for instance. That was recently approved for use on insecticidal bed nets to prevent transmission of mosquito-borne diseases. Hydromethylnon I recognize from ant baits. Others you may recognize from contexts that you're familiar with. But unfortunately, a drawback of metabolic insecticides is that we have mitochondria as well. Plants also have mitochondria, so there's a risk we might poison ourselves or the very crops we're trying to grow in our fields if we're trying to manage crop pests. So these insecticides are somewhat selective for insects more than other organisms, but there's still a risk that we could poison ourselves or other organisms we don't want to kill. So by now you might be wondering why we have so many insecticides that target insect systems that overlap with our own physiology. I mean, if it's so dangerous to us, like the nervous system and metabolism. Well, that's come up before, and there have been efforts to develop insecticides that are more selective for insects than they are for other organisms. And one aspect of this is targeting insect molting and growth. And these insecticides are known as insect growth regulators, or IGRs for short. So molting is something that we and other vertebrate animals don't do. So many of the insecticides that target molting have much lower toxicity in humans because we don't have the target sites for molting. In fact, these are among the few insecticides that we can actually feed to pets to get rid of fleas or treat livestock with, or even add to cisterns that contain drinking water to prevent mosquitoes from developing in them. So I'm going to give you a very, very brief overview of the process of insect molting, just to describe how these work. So insects need to molt occasionally to grow bigger. The cuticle that they have is a hard shell. It's not alive and it can't grow. So the insect needs to molt to make a new, bigger cuticle if it's going to get bigger. And several organs have to work together in concert for the insect to be able to pull this off. And there are a number of steps involved. So first, the old cuticle has to separate from the epidermis. This is called the hypodermis in this particular diagram. And a molting gel is secreted into this space that helps digest what can be digested from the old cuticle and helps the insect reabsorb some of those nutrients. A new cuticle is then eventually secreted and formed. And eventually, when the insect is ready, it starts taking in either water or air and starts flexing its muscles to create hydrostatic pressure. And this is to break what's left of the old cuticle, and so that the insect can escape and assume a larger shape. So great, the insect's made it that far, but once it has emerged, it needs to continue flexing and taking in either more air or water to expand that cuticle so that they can have a larger shape. That was kind of the whole point of molting. But eventually, a hardening process known as sclerotization will occur that makes the hard shell of the insect solidify. So this cockroach in this picture has not yet hardened the cuticle. But I want to emphasize that this molting is a complicated process, and one mistake and the insect could be trapped in its old cuticle and die, or it could be subjected to deformity. That's not going to make it very viable. And this process is largely coordinated by a series of different hormones. So there are many hormones involved in molting. I'm just going to talk about 4 of them today and a 5th that has to do with insect development. So when the insect needs to molt, prothoracicotropic hormone, or PTTH for short, is released by the brain into the hemolymph, again, the insect bloodstream. So this eventually travels to the prothoracic gland, where it interacts with a receptor that causes the gland to start producing and secreting ecdysone. This is another type of hormone, a kind of steroid hormone. So ecdysone travels through the hemolymph to the cells of the epidermis that initiate the molting process. So that whole series of events where the old cuticle is digested and a new one is secreted, that's initiated by ecdysone. So once that endocrine cascade is underway and the insect is ready, eclosion hormone, not shown on this diagram, is secreted by the brain. And this causes the insect to swallow air or water to expand its body, or use just coordinated muscle action to crack that old cuticle, like I mentioned on the last slide. So the insect can then exit that old cuticle. And then also, as I mentioned, once emerged, that cuticle is pliable. The insect needs to expand that so it can have a larger body shape. But eventually that hardening process will occur. And that is set in motion by the hormone known as bursicon. So this is a very simplified version of molting. Like I said, there are a number of other hormones involved, but I'm keeping it pretty basic for today. But I just want to emphasize again, this is a really vulnerable time for the insect. The cuticle is soft for most of this time, and it can't really escape predators very well during this process. Also, if it gets knocked over, it could get stuck and die inside the old cuticle. But probably the biggest vulnerability in this whole process is that there are many hormonal steps involved that need to happen at just the right time and in the right order. And disruption of these, just one of these, could be fatal to the insect. So here are some examples of molting disruptors. Some are mimics of ecdysone itself, such as the diacylhydrazines, tebufenicide and methoxyfenicide. There are many other fenicides along this list. But there are also some chitin synthesis inhibitors. So this is disrupting that process of hardening the cuticle that I mentioned. This includes some chemicals such as lufenuron. This is used in flea control for pets. A final one that I'll note is cryomazine. This is more specific to dipterin, that is fly molting. And this is a good point to, again, reemphasize that several of these insecticides are safe enough to give pets as a drug so that insects can feed on those pets and get poisoned by it. So generally pretty safe for mammals and other vertebrates. I mean, I shouldn't make too big of a blanket statement, but generally IGRs are much safer than nerve and metabolic toxins. Oops, a little too fast there. Okay, so one other hormone I want to mention is juvenile hormone. It's not a molting hormone, but it is important in development. So it's released by a gland attached to the brain known as the corpora allata. And as the insect grows, it generally molts into the same form as it just was, the larval stage. But eventually, that insect needs to molt into the adult stage so that they can reproduce and continue their life cycle on again. So, how does the body know when to make adult tissues and structures? These are quite different from larval tissues. Well, the answer is juvenile hormone. Now, while the insect is developing through larval stages, juvenile hormone, which we refer to as JH frequently, it's produced and released. So high JH titers preserve larval characteristics into the next molt so that it stays a larva. But if the JH titer ever lowers when the insect is about to molt, the insect either develops into an adult or a pupa that will become an adult in the next molt. So put another way, JH suppresses adult characteristics. There are several forms of JH shown here, and different insects use different ones. Unfortunately, I'm going to have to skip over a really cool story about the history of JH discovery and the discovery of some natural analogs, but researchers have discovered JH analogs produced by plants, for instance, and we've been able to look at those structures and develop chemical synthetic versions of those that are effective on insects as well. And some of these different JH mimics affect insects differently. So you have some insect group specificity. But here are a few examples of JH mimics or receptor agonists that are in use today. So methoprene is a classic one. It was actually developed by a former UNR professor, so we have some local claim to fame, Dr. David Schooley. But this is one that can actually be applied to water for mosquito larval management. It can be also fed to cattle to manage fly pests that lay their eggs on the cattle dung. So another one that is relatively safe for humans and other vertebrates, although it may have impacts on invertebrate life. But here we can see some mosquitoes that have been poisoned by methoprene. They're really confused developmentally. They can't quite figure out if they're larvae or pupae or adults. But there are a few other mimics besides methoprene, phenoxycarb, pyriproxyfen. And before we move on, I just want to emphasize as a group, IGRs are generally safer to humans and pets and livestock than many other insecticides because the mode of action is something that vertebrates, such as ourselves, don't have. We don't molt. But these insecticides do have their limitations. They generally only impact the larval stages of the insect we're trying to manage. So timing is a really important consideration for how these insecticides are used. If the insects have already reached the adult stage, they're unlikely to be effective. Okay, so I want to move on to physical toxicants. These are chemicals that block some important process indirectly rather than through a chemical mode of action. I'll kind of describe what I mean by that when I describe the action of two major examples. So one is an example of using insecticidal oils to manage mosquito larvae. So mosquitoes develop in the water and they breathe through these little tubes on their back end known as a siphon. It's kind of like a little snorkel that they put up to the surface of the water and they can breathe through. Now, normally life underwater is not a problem for them, but if you add a little light oil to the surface of the water, this can block those breathing tubes and the insects end up getting drowned. Okay, another common physical toxicant is abrasive dust. So I was talking earlier about how the insect cuticle or exoskeleton it is protective to the insect, not only from a physical standpoint, but it also prevents the insect from dehydrating. So the insect cuticle has a thin layer of wax and hydrocarbons and other lipids over the surface of it to prevent the insect from drying out. Because again, insects have a very high surface area to volume ratio, which makes them susceptible to drying out. So this layer is really important to protect them. But insecticidal dusts abrade or they make scratches in the insect cuticle. And through these scratches, the insect loses water and begins to dehydrate to death. So some examples of these, as noted in mosquito larvae, a light oil might be used. This would be an example of an asphyxiant. We're choking the insect, suffocating it. Now, light oil might be applied to the surface of a container of water. Typically, this wouldn't be used in a larger body of water like a lake or a pond or something that has wildlife. This would be more of a use for something like a decorative fountain. I want to make some caveats to this, of course. I don't want you to rush out and pour oil into your bird bath or something. But because obviously this oil can have contamination issues on the environment. But I am just using it as I wanted to have this presentation geared on how insecticides work rather than direct recommendations for insecticides. And that also doesn't mean I'm directly advocating for the use of any particular brand of insecticide that I might be showing on these slides. Okay, so light oils might be an example of an asphyxiant. For desiccants, there are a couple different options. Diatomaceous earth is commonly used as an insecticidal dust. Also, boric acid is sometimes used. But these are often used in urban pest situations. They're not so useful out in the field just because when they get damp, they lose their effectiveness. But for cockroaches, for instance, you can spray this into little cracks and crevices that might be harder to reach otherwise, so that the insect comes into contact with this. The desiccants get spread all over the body, and it starts abrading the cuticle, normally through parts where the insect's body that are going to be touching each other, such as the legs. These abrasives will start introducing scratches into the cuticle, and the insect dehydrates. I mean, you can kind of see this. This is a electron micrograph of some diatomaceous earth, and there are a lot of sharp edges on here that just grind into that waxy layer on the insect cuticle and causes them to dehydrate. So that's just a couple examples of physical insecticides. There are some more that I don't have time to get into today. Okay. So now we'll move on to another mode of action, stomachic insecticides targeting the digestive system. As mentioned earlier, most insect damage is caused by their feeding behavior, and the insect clearly isn't going to be able to survive if it can't eat or get needed nutrients from the food it eats. So going back to our poster, you'll note there aren't many classes of insecticides targeting the midgut here in orange and brown, but that doesn't mean they aren't important. And I particularly want to highlight this one, popularly known as BT. This is from a bacterium, Bacillus thuringiensis, that makes delta endotoxin. Now, this particular bacterium can be ingested and produce the toxin inside an insect's gut. But even further, plants have been genetically engineered to produce this toxin independent of the bacteria, so that insects are poisoned when they feed on the plant. And this has become a huge agribusiness asset to formerly Monsanto that was acquired by Bayer. So the way this toxin works is that it's activated by high gut pH that occurs in certain insects, particularly Lepidoptera, that is caterpillars. That's very different from our physiology. We have a very acidic gut in our stomach, and many other insects do as well. But anyway, once this toxin is active in the insect, it attaches to certain proteins on the insect midgut. Now, humans don't have these proteins, so we're not at risk to being poisoned by these. And not even all insects have these particular proteins. But this toxin, when it starts attaching to these particular gut proteins, it chains itself together to essentially form a hole in the midgut. And you can imagine this is a pretty big problem for the insect. Digestive enzymes from the gut can start flooding into the body cavity and start digesting tissues and organs. That's going to be a problem for the insect. But even if that damage is partially contained, some bacteria may enter the body, which ends up being a much bigger problem for the insect. It ends up dying from septicemia, infection, in other words. So, delta endotoxin is the primary example. There are some variations on this based on some other related bacteria. There are also several Bt crops, as I noted, including cotton, corn, soybeans, just to name a few. And again, this is where the plant is actually making that delta endotoxin as a result of genetic engineering. But there are other stomachic insecticides, including inorganic compounds such as boric acid. A very simple cockroach or ant bait is to mix up sugar water with a boric acid solution. This causes damage to the insect midgut when it's ingested. Now, it's not quite the same way that Bt damages the midgut, but again, this is probably not the main mode of action either. It's damaging the midgut, but it may be rupturing the gut to the point where bacteria or digestive enzymes can get into the body cavity, and the insect can die through bacterial infection. I also want to note here, since we're talking about stomachic poisons, there are some microorganisms that infect insects through the gut and end up killing them. So they're pathogenic to the insect. They're insect diseases, in effect. One example is Nosema losticae. This is a type of protozoan that infects grasshoppers and crickets. Now, this is a living organism, so the mode of action is rather complex, and as it attaches to the insect's gut, it infects it and invades the insect immune system, or invades the insect and has to avoid the immune system, and eventually makes the insect sick so that it dies. But the mode of entry, at least initially, is the digestive system. And there are other stomachic insecticides that I'm not going to have time for, but I really wanted to include a section on the action of biological organisms such as Nosema locustae, but not time for it today, unfortunately. But I also wanted to mention that there's a number of insecticides that we don't know yet what the mode of action is. You know, we have some ideas based on how the insect behaves before death or what organs seem to be damaged, but the mode of action is still somewhat unclear. And I just have a very brief list here. It doesn't include all, but sometimes an insecticide was discovered in a way that doesn't clearly suggest how it is killing the insect. And azadirachtin is an interesting one. It's derived from the neem tree. This is a tree that's native to Southern Asia, and it was noted for its ability to withstand locust swarms. Now, you may have seen or maybe even experienced locusts coming into an area and just devastating all plant life. There are just so many of them. They're so abundant. They tend to eat all plant life, and then they fly onto a new area. But what was notable is that the neem tree was largely unscathed during these locust swarms, and people noticed this and eventually started purifying extracts from these trees, and azadirachtin was discovered as a result. But despite that, it's still unclear what the mode of action exactly is, and there may be multiple modes of action it's impacting in the insect's body altogether. Oil extracts or just other types of extracts from other plants are another example. You know, again, in some cases we have hints that what the active chemical is doing, but confirming a definitive mode of action is still in process. You know, we know it kills the insect somehow, but it's not quite clear how. Okay, and going back to our list of organ systems, there are many other organ systems and tissues that are as yet untapped targets for new insecticides. Perhaps we can interfere with reproduction. You know, pests aren't going to be a problem if they can't lay eggs. Or maybe we can target the excretory system. If the insect is unable to get rid of wastes, eventually those toxins are going to kill them, and so on. And some of these systems have, in fact, been targeted for new insecticides. Some are in development. But all of these are potential weak points in insect physiology that we can exploit. And another thing I want to note is that it may not always be necessary to directly kill the insect. Perhaps if we can just deter them from feeding, they might leave the crop field, and that's really enough. So behavioral modification, in other words. So I think that's a good transition to talk about what I work on, which is insect neuropeptides. These are molecules that may serve as neurotransmitters or hormones in insects. As you might have noticed from earlier in this talk, neurotransmitters are an important target at the synapse. Also, hormones are an important insecticidal target. Now, the insect I focus on is Lygus hesperus, the western tarnished plant bug. This is a pest in a lot of different crops, most notably cotton, strawberries, and alfalfa, but there are many others. And one particular set of neuropeptides I'm working on now is insulin-like peptides, or ILPs. Now, you might be familiar with what insulin does in our bodies. It helps us regulate our blood sugar. We've shown that ILPs and Lygus hesperus also have this function, but we have evidence that they may have other roles as well. Particularly, we're interested in the role of ILPs in reproduction and diapause. Now, diapause is the term we use for when the insect is in a dormant state, such as in the wintertime. It's roughly analogous to hibernation. But disruption of any one of these processes could be detrimental to the insect. If we disrupt blood sugar levels, for instance, the insect could die. If we disrupt reproduction, the insect could be unable to mate or lay eggs. If we disrupt diapause, the insect might not be able to survive winter, and we'd start the spring with a pest-free field. So there are a number of potential benefits to this strategy. And beyond ILPs, there are a number of other neuropeptides in insects that could be targeted. And one approach we're taking to this is borrowing a page from drug discovery by expressing the receptors for these neuropeptides in reporter cell lines and expressing those in reporter cell lines. These would be cell lines that might glow, for instance, when the receptor is activated. And we can screen these in 384 well plates against chemical libraries to see which chemicals activate these receptors or prevent them from working in the first place. These chemicals then can be counter-screened against vertebrate receptors, receptors in our bodies that we don't want to damage, to avoid wasting time on broadly toxic compounds. But the chemicals that make it through that screening process can eventually be tested on the insects themselves. And the effective ones, we can look at the chemical backbones and see how we might modify that to make them either more toxic or more specific for the insects we're actually trying to kill. And eventually, we want to partner with private industry to help bring these insecticides to market.